Okay, we are recording this webinar and we do expect to post it to our YouTube channel in the coming days. I will send out a message to let everybody know that it's there. So some people will continue to join us tonight. I know some people told me that they couldn't make it tonight, but they'll watch the recording. So we are happy to be here and happy to provide this for you. So tonight's webinar is Bioacoustics for Scientific Research. And David, if you could go to the first slide, the next slide for me. There we go. So this webinar does allow us to take questions. It's not like super straightforward. So here's a little bit of instructions. You can send us a reaction or you can ask a question. And I think this is for the website. David, if you go to the next slide. This is what the app looks like, the Google Meet app. So feel free to ask us questions when David gets done. Hopefully he'll answer some questions along the way that you may have posed when you signed up for the session. And then when we get all done, he will probably have time to answer some other questions as well. So looking forward to getting those questions from you as we go along. So let me introduce David Mann. David, if you want to go to your first, there you go. So that's a little bit more interesting an intro. So David is the founder of Haikubox, and his background, he's spent decades studying sound production and hearing in all kinds of animals. He'll talk about some of that tonight. But he got his PhD from MIT and the Woods Hole Oceanographic Institution. So his first critters that he studied were fish. And then he's done lots of other things. He was looking at sound production. He went on to study hearing of fish as a postdoc at the University of Maryland in College Park. And then he joined the faculty of the University of South Florida, where he expanded into other animals and was doing a lot more sound production rather than hearing of animals. He founded Loggerhead Instruments. We still run Loggerhead Instruments, and he'll go through how Haikubox was started, but we still sell underwater equipment, underwater recording devices to researchers and consultants and people all over the world who are studying underwater sounds. So that's the primary focus of our business, and Haikubox fits in that quite nicely. I do want to, before we start, thank our sponsor, the National Science Foundation, because we're a teeny tiny little company of 4 people. And without their support, Haikubox would never have happened. So we are grateful for their continuing support. And we hope that there'll be funding for lots of other activities by the federal government in the future. So we're really grateful for what they've done for us. So I'm going to pass it on to David, and I'll look forward to seeing your questions, which I will bring to his attention at the end. So thank you very much.
Okay, thanks, Amy. Can you hear me okay?
Yes. Thank you.
So I'm gonna talk about somewhat the history of bioacoustics, and a lot of it's from my personal perspective and things I've been involved in. But what I want you to come away with is an appreciation of how rapidly things have grown in the past even 10 years from what they were back in the early days. So I was all of 3 years old when Songs of Eastern Birds did not make the top 10 charts of Billboard, but I did get a copy when I was like 6. And so as a kid, I was really always into the outdoors, kind of went bird watching and trespassing all over the place as a child. And then I guess I grew up. But back in the early days, Jacques Cousteau in 1953 had a book and a movie called The Silent World about the underwater world. It's anything but silent. Jacques Cousteau knew this, but I guess it was a good title. So let's see. So a lot of understanding about bioacoustics comes down to there's two parts to it, right? There's the recording of the sound, and then how do you visualize the sound? So early days, you would have perhaps musical notation of like, this is what a nightingale sounds like or a cuckoo sounds like, but you can't quite capture it. And so that's where the spectrogram comes in. So for a lot of birders, right, you have mnemonics for a sound that a bird might make. So here's a tufted titmouse. Right, so that's a clip from a Haikubox, and it sounds like Peter, 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 if you're English speaking, probably. But that doesn't really capture everything about the sound, and that's where a spectrogram comes in. And I had talked about this in the previous seminar, but I wanted to go over it again, so in case you weren't there or you want a little refresher. And so a spectrogram is an image of sound, and it's got time on this X, this long axis on the bottom, and then frequency or pitch is here on the Y, or the vertical axis. So anything that appears, let me see if this works. Anything that appears as a horizontal line like this is going to be a tone. So that would be more like, if I can make that yellow. Right, so this part, that's a tone. Anything that's going like this is going to be it's decreasing in frequency as you go down. So if we listen to this one more time, back over here. So we're clearly hearing this part, right? But this transition part, I don't think we hear that well. And actually, if you look carefully, there's also this kind of echoey part at the end. So a spectrogram is capturing a lot of detail about that sound that's not captured by musical notes or by simple mnemonics. There we go. So, so back in the early days, so recording of sound, right, started much earlier. Thomas Edison and a lot of innovations with magnetic tape, but the actual visualization of sound came much later. It was largely driven by developments during World War II. Largely aimed at kind of encrypting, decrypting human speech, but it was also used for other things as well. After World War II, a lot of the tools that got developed got used by scientists. So this is Ellerby Davis, Cornell Lab of Ornithology, using a device called a K sonograph, and it was a purely analog, no digital, analog tool for making a spectrogram from a recording. So what you would do is you would have your tape that you recorded your bird on, and then you would play it to this machine, and this machine had its own magnetic sort of tape inside of it that could hold a few seconds of sound. And then what it would do, it would play that over and over again. And if you look here, there's this drum in the middle, and that had temperature sensitive paper on it. And there was a heating lamp that would traverse this and basically burn the paper. And so they would basically have a whole bunch of filters. So it would be like very low frequency filters to get the low frequency sound as it spun around. And then it would switch to a higher frequency filter, and then you get the next layer on the paper. And you can see he's looking at one of these printouts here. And these are just a few seconds long. Okay, so this took a lot of time. And if it didn't work out, you had to redo it all over again. And I just want to contrast. I was just thinking about it because I was talking with Amy earlier today about the amount of data that we have coming in. And so with your Haikubox, it's actually making a spectrogram on the device every 3 seconds continuously. And that is basically nothing we could have dreamt about in the 50s or 60s. Even when I was in grad school, so this is kind of early 90s, the first computers were kind of able to start doing spectrogram generation. And the early ones would take tens of minutes to actually calculate it. Things have progressed so fast and gotten so small that pretty inexpensive devices can now just do these things really, really fast. So, you know, we're taking in like 10 million a day or so. So even in one day, we probably exceeded all the spectrograms printed on the K sonograph ever. It's crazy to think about. So, so this is kind of one of the first papers that showed how sound could be used to study the ecology of animals. And this is Charlie Breeder, who's kind of my academic great-grandfather. And so he was at the American Museum of Natural History in New York City. And he would come down here to Sarasota, where Haikubox is based. And he was studying this fish called the oyster toadfish. And it makes, I don't have a recording here, but you'll hear one in a sec. He would sit there with a microphone wrapped in a condom, stuck in the water, and count out when he heard these fish. And they call it night often. And so he would just go down there at dusk and make these counts. And he did it over a year-long period. And what you could see is in the winter, November, December, January, February, he wasn't hearing very much. And then as the water warms up, the fish get calling. So basically in the spring, they get calling a lot. And then in the summer, a ton. And this paper was actually pretty influential for my thinking when I was doing graduate school starting out, because I was like, oh, you can see like the fish that you can't even see, you can kind of tell what it's doing over a really long period of time. So this is the sound of the Gulf toadfish. So it's making this boop sound. And so on the spectrogram, sorry, I'm switching screens here so I can draw on it. The toadfish call is right here. Let me switch to white. So it's a line right here. And there's 3 or 4 of them above each other. And so what it's doing is inside its body, it has a swim bladder, which in most fish that have swim bladders is used to regulate buoyancy. So, you know, they don't sink. They don't have to keep swimming to stay up. But the toadfish has evolved a set of muscles on the side of the swim bladder here. And those muscles rapidly contract to make this sound. So this sound might be at the fundamental at 200 cycles a second. So it's contracting those muscles 200 times a second. And so they're among the fastest contracting vertebrate muscles out there. And what you're hearing is basically this swim bladder changing size and producing this what's called a boat whistle call. Here's a different fish. This is red grouper. It's in the Gulf of your choice of words, and also in the Atlantic Ocean. And they're a common fishery species for grouper sandwiches and things like that here in Florida. So this is when I was at University of South Florida, and we were trying to identify sounds produced by them. And so this is about 100 miles off the west coast of Florida. There's a whole bunch of marine protected areas where fishing is not allowed, and which are areas where these red grouper are spawning. So they set up territories, and they spawn here. And so they're protected, and we're trying to figure out technology that we could use to better figure out where they are, because you're basically relying on catch data, which isn't really going to help you that much in a marine protected area. So we actually use, this is the Liberty Star, which is one of the 2 vessels that NASA used to use to recover the solid rocket boosters from the space shuttle. So we took the Liberty Star over to the Gulf of Mexico. This is Felicia Coleman, and with her husband Chris Koenig at Florida State. We had an ROV on the bottom. So this is about 240 feet deep, which is deeper than you can dive on regular compressed air. And so we have divers who are basically doing decompression diving with us, and they're putting cameras down where these fish are. And basically the reason we had to use this vessel is the vessel could hold its position without anchoring. And so these guys are basically running an ROV to see what the divers are doing, and to communicate with the divers from the vessel. And so we just built these off the shelf. We actually used race car cameras that we put in these, and then this is our underwater microphone called a hydrophone, and then they're just on tripods pointing at these pits that the fish dig out. And so this is 2008. And so if you could hear it, it was like a boop, boop, boo. There's 2 fish in this. The one, and I'll play it again. This fish with the bars, so these vertical lines, is the female, and the male is this one back here. So basically she's coming in to check out his little home he's dug out, and he's trying to impress her with his sound. So this was actually the first time we stuck these recorders out there where you're able to get this recording. The part of the point of showing you this is to show you that there are a lot of other animals besides birds making sounds, and some of them are in places that are extremely hard to study, hard to access. But once you know this sound, you know it's red grouper, and so everywhere you hear it, kind of like with Haikubox underwater, you can figure out where the red grouper are and what they're doing. So again, here's the spectrogram. So it's whoop. So whoop, whoop, boo. Okay. Okay, so that's very low pitch. If you're listening on a phone, you actually might not hear it. You kind of usually need headphones or laptop speakers. So now that you have the sound, one question is, so how do you figure out where all of the grouper are? And let me skip that. And so one tool that we have that's been developed in the kind of early 2000s is an underwater glider. So what it does is it doesn't have a propeller on it. It basically changes its buoyancy to glide up and down in the water. So kind of like if you were in a pool and you breathe out, you sink down, and if you breathe back in, you rise up, and you're just changing your volume. So you're changing your buoyancy. And so they are very efficient. They can run a long time, like weeks, on not that much power, and then they're really slow. So they're moving like 1 to 2 miles an hour tops. And so we ran one. This is our first glider deployment. This is 2009. So this is the west coast of Florida, and the glider was dropped off here. So this is about, the water depth is about 100 feet, and then we run it out to where it's 150 feet, and then we come back. And so we basically built a recorder to stick on to the glider to record what we heard along this path. And so a bunch of things were recorded, and we don't know what they all are. So I'm just going to play them for you. Okay, so if you can hear this one is like woo. Okay, so you have, again, if you look at the spectrogram, it's this line going across, so it's kind of single tones. As soon as you have these harmonics, like 3 of them, it starts changing from a tone like a to more of a you'll see that or hear that sort of thing in the nuthatches. If you look at spectrograms of nuthatches, they kind of sound like they're honking. That's when you have these harmonics in there. So this fish is unknown. And it was detected basically everywhere these light blue dots were along this path. So it's all over the place in the Gulf of Mexico. Here's another one. Make that louder for you. Okay, so it kind of sounds like a horn or kids fooling around. We don't know what this one is either. And if you look at where it was heard, it was mostly in the deeper water. So these are like these yellow dots. And then we also heard red grouper, which we listened to before. And they were found somewhat like closer to shore, and then a lot more of them farther off. And so again, they weren't rare in this one spot. We just ran the glider for the first time. One thing I wanted to point out is you might notice that there's banding here in some of these. And so like this light blue one, and what's happening is, the glider is only going like a mile an hour. So going really, really slow. And so this might be daytime, and now it's nighttime, and now it's daytime. Sorry. So here, change color. Right, so here's nighttime, and then it's daytime. Those fish stop calling, and then it's nighttime again, and they're calling again. So even though you don't see this one going wonk in here, doesn't mean they're actually not there. If you look at the return trip over here where the glider came back in, you see that that fish is present. So the glider has this, there's this trade-off between you're not everywhere all at once, right? So your sampling is spatially and temporally correlated. Okay, so closer to shore. So we're going to move in, and I'm just going to play some sounds from Sarasota Bay. So that's a small bay here on the west coast of Florida, south of Tampa Bay. And you can hear a lot of things going on. So you'll hear like this crackling, popping. That's these vertical lines here. So some of them are snapping shrimp. So if you look down here, let me switch to white. So these vertical lines, anything that's a vertical line in a spectrogram, it's very short in time and has a lot of frequency in it. And so those are clicks. They're like snapping your fingers, clapping. That's going to be this very broadband, meaning from low frequency to high frequency, but very short signal. So there's snapping shrimp here, but there's also dolphin echolocation. And so just if you can see this scale bar here, this says 20,000. This is the limit of human hearing. So we can't hear anything above this 20,000 line. The dolphins are echolocating at 80,000, 100,000 hertz, and their hearing is much higher frequency than ours. So they're able to use echolocation to navigate and find prey. The other thing you'll hear is this whistling, right? So you see this line kind of going up and down. So I'll play that again. And again, that's pretty high pitched. So like even So as we get older, like I'm probably not hearing the upper parts of this, but that's a dolphin making a whistle. I think I forgot to say, so these low frequency snaps are from snapping shrimp, which is this tiny, tiny little shrimp that has a claw, and it basically closes it really fast, which generates a cavitation bubble that collapses. So what you're hearing is actually collapsing cavitation bubbles that this little critter makes. So there's a lot going on. It's very cool. And what I want to talk about is kind of how we can unpack that to understand what's going on in this environment. And so this is work from a colleague, Randy Wells, and a bunch of his collaborators who have studied bottlenose dolphins here in Sarasota for over 50 years. So they have a fin catalog where they can recognize individuals by the shape of their fin. So sometimes they have little nicks and notches taken out of them. They can tell them apart. But even more amazingly is they now know who is related to who. So if you look here, this is in 2007, this is a calf. So this is younger, nice clean fin. This is that calf's mother. So they have science names like F125, but I believe a lot of them have more fun names that don't get published. This one is the grandmother. This one is the great-grandmother and great-great-grandmother. And I don't know, so if Randy were here, he would tell me like if this one actually had a kid. So multi-generational knowledge about what these dolphins are doing. If you look at their distribution, and so if you look at the calf, which are these red dots, it's living kind of more down here in the main part of Sarasota Bay. But if you look at, let's see, so yellow is FB37, that's the great-grandmother. She was hanging out up here, okay? So there's been a shift in the population of this family kind of to the south. So that's all from photo ID and a lot of work by a lot of people out on the water. But these dolphin whistles are pretty cool in that they have a type of whistle called a signature whistle. And so that whistle is unique to that individual. And so this is work by Peter Tyack and Layla Sayag, who were at Woods Hole Oceanographic Institution. And so they recorded the signature whistles of these dolphins. And so this is FB54, so one dolphin, but from 1984 to 2006, you can see the shape of the whistle is pretty much the same. And then here's FB157, same thing, very different whistle. Like you and I could just look at that and go, oh, yeah, I can find that whistle. That's this FB157. So this is really cool because we know a lot about these individuals, and Layla and Peter have a catalog of all of the individuals' signature whistles. And part of the reason I'm talking about this is someone asked if we could actually tell individual birds on the Haikubox apart by their sound. And we haven't tried yet. I think it would be difficult for most, but I don't know if it's impossible. So it would certainly be something worth looking at. So we're building with a bunch of collaborators, Randy, Katie, Athena at New College of Florida, and Peter Simard and Shannon Gowans at Eckerd College up here, a network of listening stations that are essentially running like Haikubox, like firmware on Haikubox, like hardware, but they're listening for dolphins. And so there are stations that people have in their backyards or they're in parks, and we have a hydrophone, so an underwater microphone running over the seawall, and then those are listening and recording continuously. So just to give you an idea of kind of what we can get from this sort of station, this is August 2018. And you're going to hear, well, it's easier to see. You can see a dolphin echolocating. You can see a lot of these snapping shrimp calls. Sorry, go back. And then there's down here, there's a fish calling as well. That's all this low frequency stuff. So that's one 10-second spectrogram. Now, if you looked on the 1st of September, it's really quiet. So this is playing now. There's one lonely fish down here. So here's a fish calling, and it's gotten super, super quiet. All the sampling shrimp are gone. And so what's happened is, so here's a little social media post, but basically red tide has come through Sarasota Bay. And red tide is a microorganism that produces a neurotoxin that is toxic to fish and invertebrates. And so this is just looking at sound levels over time, but you can basically see the calling, like August 7th to August 9th, 13th, just keeps getting quieter and quieter and quieter. So you're able to see the effect of this red tide event on the bioacoustic signature of Sarasota Bay in real time. So what we're trying to work on next is actually using AI to not only detect the dolphin whistles, but then to group them to individuals. So, okay. Now on to birds. Okay, so in the early days, right, we had bird songs on records, and we got bird songs on tape and bird songs on CD, and then we got eBird and Merlin and the Library of Natural Sound from Cornell. This is, on the right here is BirdNet, so it's similar to Merlin. It's an app made by Stefan Kahl, who's working with our colleague at the Cornell Lab of Ornithology, Center for Conservation Bioacoustics. And you basically can use it like Merlin. You can run it in real time, but then you actually manually go, oh, I want to know who made this sound. You can select it, and it will run it through BirdNet to identify the species. So what it's doing, so I'm going to give kind of an overview of how it works, and then I'm going to go a little deeper, but not too deep. So the microphone is recording the raw audio, and then the metadata, which is basically where you are in the world. And then it's generating these 3-second long spectrograms, and they're overlapping. They're like 3 seconds, and then they'll shift a second and make another 3-second one. And they're run through the BirdNet algorithm, which was developed by Stefan Kahl. So he was he's a scientist in Germany, but he works for Cornell, but also at the University of Chemnitz. And BirdNet was the first really good bird identifying algorithm. And what it does is, for every species in the algorithm, so the first version he made had about 1,000 species that covered the US and Europe. For each one of those 1,000 species, it's going to give you a confidence score. So it might say, oh, 91%, that's cardinal, 14% is this other species, 9%, right? It's European goldfinch, right? And then there's a eBird occurrence mask. We refer to it colloquially as the grid. But basically it says, where I'm standing, am I likely to be hearing a northern cardinal or a European goldfinch? And so it's basically filtering the detection's confidence scores by a species list for where you're standing. And just assuming you have a Haikubox and it's missing a bird that is really obvious to you, just email us, because our species lists aren't perfect. They're getting better. So, but if we're missing a species, we will never show it to you. We actually still detect it and store that information, but we're not showing it. So if it's missing something that's obvious and you know it's there, let us know. Okay, so that's kind of the overview of how it works. Someone had asked, what is actually happening inside this neural net? And so the type of neural net that's being used is called a convolutional neural net, which just means it's running different types of filters over the audio recording and extracting different features from which it's using, which it's making the identification. If you think about, so I'm gonna give you an example in the visual world because it may make more sense, because by the end it's going to make no sense. Okay, so this is from this website showing basically how you could look at what's going on inside a neural net that's been trained to identify different types of images. So this one, this neural net has been, I don't know if it's just trained to recognize like birds, cats, dogs, or different species of birds. But in any case, if you feed an image of an American robin through it, what you get after the first layer are 64 different representations of this image, right? And you'll notice they're all black and white. And they focus on different features, right? So if you look at this one here in the upper left, like here, you'll notice it has just the outline of the top left part of the bird. And that's really what you're seeing. If you look at this one, it's looking at just the silhouette of the bird. And then if you look at the one down here, it's highlighting the orange breast part of the bird. Okay, and you can see that orange breast part of the bird appear in a whole bunch of these different representations of this image. And so what it's doing is the net has been trained to recognize these images. The training process involves creating filters that are going to filter the image in lots of different ways to try to extract features that are important for making the recognition. So this is the first layer, and this is the layer that's going to make the most sense, because as you go, so these are multi-layered things. So these images are then going to get fed into the next layer of the net, and you'll start, a lot of them start not even looking like birds anymore. And then if you get down to the last layers of the net, you end up with something like this, right? Which is totally unintelligible to you. But the images have become a lot smaller, but they're basically highlighting the things that are unique about an American robin in an image. And then it's taking all of this information and running it through a fitting model to come up with the confidence score that that's a robin. So that's what's happening. Another way you can think about it is, even if you're looking around, like right now, your desk, your computer, and think about what your visual system is doing. One of the things that we're really good at is finding edges of things in our visual cortex. And so like the edge of the screen, the, whatever, the coffee mug that's sitting next to it, we're really good at picking out edges. And so often these nets get really good at finding edges and things like that as well. So let's see. Okay. So if you think back to the beginning, right? We started with, oh, it's going to take us hours to make these spectrograms burned down to paper, and then what are we going to do with that? And that's assuming we got the recordings anyway. Now we're to these really large scale projects. So this one here is being run by the University of Wisconsin and Cornell Lab of Ornithology, and they're very interested in studying the California spotted owl, because there aren't many of them left, and a lot of their habitat is going away. And so they put out 1,641 audio recorders in 2021, which is just a huge task. So if you look at this part of the image, you can see where they are in California. So in California, this is a huge, huge range, right, that they're covering. So it's zoomed in on all of these plots. This paper is basically, they'll put like 1 or 2 or 3 in a hexagon, but they're really close together. So this paper just came out. It's at the bottom is the citation, McGinn et al. 2025. Ecological applications came out last month. And so what they're trying to do is look at what is the effect of forest fire on owl populations in the Sierra Nevadas. And they looked at 6 species. I'm just showing you 3 here. And so they're basically looking at occurrence in these hexagonal squares or hexagons. And this is just for the spotted owl detections. And if you look, so the North Complex fire, so this guy up here, right, was in Northern California in 2020. The dark red areas are where there was severe fire, and then the orange areas were low to moderate severity. Where there's a hexagon that is a solid, like a bold shape or color, they have detected the spotted owl. Where it's gray, they have a recorder, but it never picked up a spotted owl. So what you can see from this is these dark red areas. I mean, this is only a year later. They haven't really moved back in. But this one here, the King Fire B, was in 2014, right? So these recorders were put out 7 years later. Where it was severe, the spotted owl has not moved back in. You can find spots where it's less severe, where they have detected spotted owl, but basically they're not moving back into this habitat. So this is pretty amazing. So, and not all, just if you go look at the paper, not all the owl species had the same response. So some of them actually would go back in. But it's basically helping to predict what's going to happen if you have, you know, a very severe fire or a low or moderate fire on the ecology of this endangered owl. Okay, so Haikubox. So the reason Haikubox got started was I actually, so I was friends with Holger Klink, who's the director of the Center for Conservation Bioacoustics at Cornell. So he is a marine bioacoustician as well, so that's how I knew him. But I said, you know, we're thinking of trying to do some sort of consumer bioacoustics product to expand beyond the underwater development we were doing. And that's when he mentioned that they had basically just invented BirdNet. And so we went through tons of different iterations of design, but we basically that our connection, you know, personally is kind of what led us down this road. So,
Hey, David, just a reminder, you have about 15 minutes left.
Okay. So assuming most of you have haiku boxes, right? This is the typical display. You'll see different species that you get, and you can listen to their calls. And one of the reasons that, so there's a couple of reasons we started Haikubox. One of them is just like to understand the scale of what is going on with different birds. So if you look at the like typical range maps that you would see for a bird like the black and white warbler, it'll show you where you might expect to see them year round, where they would migrate, where non-breeding regions are. But as you know, things are getting warmer, except sometimes they get a lot colder right now. And this is a range map predictions that Audubon has made for the black and white warbler. And so you can see, so blue is the winter range and this yellow is the summer range. And you can see it's basically shifting up. Both the winter range is shifting up in the southeast US, and the the summer range is shifting way up into northern Canada. And so this is a prediction, but we want to be able to actually try to measure this and see how it happens and be able to do that on a very fine time scale, right? So like right now, the kind of polar vortex is sort of weak and the middle of the country is got really, really cold. And it's like, does that have an effect on what the black and white warblers are going to do next year? So the idea is like we could build a very large array of haiku boxes to listen to find out what's going on. We are getting lots and lots of data. So in January, we just took a look at our database and we have over 1 billion recordings. This sort of distribution of species, like 5 species make up the top third. This has been pretty stable over the past few years. They kind of change position about who's the most common. But this sort of distribution, 5 is the top third, the next 15 species is the next third, and the next 510 is the bottom third. So one of the things that we're doing with the data is, we're trying to figure out what sort of scientific analyses we want to do with it. And so this past April, if you remember, we had the solar eclipse that ran from Texas through Arkansas, Ohio, New York, Maine, and Canada. And so we thought, oh, this is going to be a perfect opportunity to kind of just test, what can we learn with the Haikubox. So if you look at this, the eclipse was pretty fast, right? So this is down here in Texas. This is 1:30 Central Daylight Time, and by the time it gets up here in the Northeast, that's around 3:30 Eastern Daylight Time. So it's moving really rapidly, and also in a very narrow band. I think I might not be telling the truth. Yeah, and when you're outside of this band of totality, it's not super, super dark. Okay, so the people that really saw, like, that were in the center of this, they got, a few minutes of totality. So we got ready ahead of time, and I figured out like which haiku boxes were in the path of the eclipse and said, okay, we're going to analyze these, and within 2 hours after it, we're going to know what happened. And so this was our first analysis, and everything is relative to the peak of totality. So that's what this 0 is here. That's what is the darkest. And then so over here to the left is before the eclipse, and then here to the right is after the eclipse. And this is the total bird calls per minute summed over all of those haiku boxes. And so you could see on April 8th, which is the day of the eclipse, there was this huge drop in call rate. So we were really excited. It was like, oh, this is what everyone said would happen. Yay, great. And then, so we push out this image. So then we started to look at this in a lot more detail, right? Because this is adding all the birds together. We don't really know what's happening at each location. So this was the distribution of haiku boxes that we had at the time that are sharing their data with Cornell. So there's a little box in your account page, and you say share data with Cornell. If you do that, you'll get used in these analyses. And so you can see the black dots are the locations along the path of totality. Okay, and then the blue ones are 95%, kind of what's called maximum obscuration. It's how much of the sun is covered. So so we focused our analysis on these black locations. Oh, there actually were more here, but we've removed some. So it turns out one thing we learned during an eclipse is a lot of people go outside at 2 or 3 o'clock in the afternoon and start yelling. And so one of the things that Haikubox does is if it hears speech, we actually don't upload recordings. So so we went and actually analyzed to find locations where no human speech was detected, and only those were included, and that got rid of something like half of them. So this reanalysis is basically looking at the Haikuboxes where there wasn't a lot of humans running outside making a lot of noise. And when you do that, that beautiful, nice graph that we had before gets a lot more complicated. But on this plot, this is the vocalizations per 3 minutes. So we just broke it up into 3-minute chunks because the eclipse doesn't last very long. But you also need a long enough period of time to actually get a reasonable number of counts. So this is the average, the black line of the ones that are in the path of totality, right? And so, so you can see, there we go. So path of totality, the black ones is this line. And so you can see this drop, and then it kind of goes back up. There's a bunch of interesting things about this drop. It occurs at the peak of totality, but also lasts a bit longer after. And then at the other sites that were not in the path of totality, there's nothing obvious going on. So, but if we look at individual Haikuboxes, right? So we know all the birds that are calling, and we just looked at the top 5 per Haikubox. So this is in Maine. So there's one or more pine siskins calling away. And then at the time of totality, right, this 0 minutes, they stopped calling. There was still this black-capped chickadee calling a little bit, and then they picked up again afterwards. Okay, so that's really cool. And I can tell you now, this is like the most obvious, best looking graph I'm going to show you, because then there comes all the other ones. We go to Vermont. So black-capped chickadee was calling the most. This one started actually calling during and after totality. There's a pine siskin here, wasn't calling before, waited to call after in totality, so that's in orange. So what you're seeing in Vermont, right? So this is one site in Maine, pine siskin stopped calling during totality. Vermont, pine siskin started calling right after. So a very different response. Down in Kentucky, we had an American robin who one or more started calling a ton as the eclipse was starting and peaking, and then got quiet afterwards. And then this is that robin in Kentucky. So this is at totality, right? So this is that recording of that robin in Kentucky at totality. So the simple explanation that we had, immediately after the eclipse, they stopped calling, is complicated, right? So different birds are doing different things in different places. On average, yeah, they called less, but it was not as simple a story as we first talked about. The other thing I wanted to point out was especially if you look at this one, anyone who experienced the eclipse would know it not only gets dark, it also gets cooler and the winds die down. So the birds could be reacting to any combination of both the light level cues plus the wind and the temperature. So very cool. So let's see. Okay, so I'm going to wrap it up here. Thank you for your time and attention. The other reason we wanted to start Haikubox is we actually want to try to make a difference. And so owning a Haikubox on its own, it's great. You learn a lot, but if you can do anything around your home to increase, especially native habitat, go ahead and do that. The birds will thank you. So thanks.
Okay, thank you, David. So I don't see any questions. Let's see. I think the way to ask a question is to look at the triangle, square, and circle at the bottom right, which says activities, and then click on Q&A to ask me something, or there's a chat you could use to ask us questions. We can stay on. So we have 5 minutes left, but we can stay on for a few extra minutes if people have other questions. The thing I neglected to say, David, is that you grew up in Syracuse, New York, and so you actually went to BB Woods as a kid, as a young birder, in addition to trespassing on people's property. But
yeah, I did. My brother also brought me there because he had a girlfriend that was going to Cornell at the time. So I didn't really grasp that.
And you and I went to Cornell too. So
and we're married. I don't know if you told that.
Yeah, for a long time. All right, so I see a question. What kind of science projects can school kids do with the Haikubox results? So I can answer that. We have some lesson plans posted to our website, the haikubox.com site, that you can share with anybody you'd like. We've given away actually quite a few haiku boxes to schools and nonprofit organizations with the hope that they will use them and teach those lessons or other lessons with their kids. And then we have actually posted a blog about it to our website, also the haikubox.com website, about a school in Massachusetts that made a gigantic lesson. So I'm a former teacher, so this gets me really excited. They did a huge school-wide lesson where they jazzed up the library and they wrote down all the birds that they identified, and they really had a good time with it. We feel that science is tough for some teachers, and it's important for everyone to be scientifically literate. And birds are very non-threatening and a great way to introduce kids to the concept of science and of observation and data collection. And so we think it's a great way to get kids involved in science, in addition to becoming birdies. So, okay, so I see another question. Have there been any efforts to create a tactile version for people who are visually or hearing impaired to be able to access spectrograms?
So we haven't done anything with that. So we have a few customers that I know of who are blind, and there's a lot I need to do with the app to make that accessible. So that was a learning thing for me. So there is on the account page a toggle that you can hit that will kind of put it into a more accessible mode for blind people, because it basically, you don't need it reading to you the image of a blue jay. It doesn't really help. So we've done that. For hearing impaired folks, so you can see the spectrograms, and I don't know. So I guess we could do things like where it's more comparative between them, but really that's what the BirdNet and our nets are doing. They're looking at the spectrograms. So yeah. So most of the work that we need to do are actually for blind people to make it better. And
I just add, if folks have ideas, they should, because we'd love to hear it. So info@haikubox.com would be great.
Just one more thing on that. Also, so like if you're blind, right, and you listen to the spectrogram, right, you're hearing the spectrogram. The thing that we don't have is, if you want to hear, tell what time of year that the bird is calling, we don't really have a good way to show you graphs, or to present graphs so that you can hear them. And there's a bunch of cool work where people are creating sort of audio versions of graphs. So, with piano noise, and it goes faster if the graph is higher and slower if it's lower. So there's a lot of like really cool stuff that could be done.
Okay, next question. I live under an airport flight path. Does aircraft noise pollution hinder bird nets accuracy?
Um, I I don't think it will impair the accuracy that much, but it will. So any background noise is going to probably decrease the range over which it's able to hear something. So generally, like you need to be able to see it in the spectrogram above the background noise. And so that said, if it's like a high-pitched, cedar waxwing calling, it's not going to matter that you're in a flight path. It's going to still pick up those cedar waxwings because they're so high-pitched. But if you have, a lower frequency owl or something calling and there's a jet going by, then it might miss it. It might have detected it if the jet wasn't there.
Okay, and I have two questions that have to do with the future.
Yeah.
One person asked, are there any upcoming enhancements to the Haikubox that are planned? And somebody else asked, what do you see as the future for use in insect calls, for example, katydids and crickets?
So upcoming enhancements. So we are working on, we're working on a bunch of things. So we're working on a Haikubox that is intended for use by scientists. So it'll look kind of like this one, sneak peek. And so it can record audio to an SD card, but it can also do detections just like the regular Haikubox does. But we're making that one so it could be solar powered because we've had several folks approach us that want to use them for science in remote locations that they're not able to use the current version of Haikubox for. As part of that, we're also working on having longer audio clips as part of it. So right now, the reason you're getting these 3-second short clips in the Haikubox app is that is what BirdNet is inferencing on. And so we're only keeping those 3 seconds. So we're on this new design, we're working on getting longer clips, which we will be able to bring over to the Haikuboxes. If you already have one, we should be able to get that working for you. Part of that, I'm not going to promise yet, but we hope to have the ability to stream live. So you could listen to your Haikubox if you're not at home or it's somewhere else. So that's two things. There's some other things. So the insects is interesting. We haven't worked on IDing them. So there's a number of things that we detect that we don't show right now. So one is like dogs barking and things. Like we don't show you the dog barks, but we count them. And then there's several frogs that we're interested in, both because the frogs are interesting, but also because they false detect sometimes. So for example, spring peepers in the east will sometimes false detect as osprey. So we're kind of working on improving that. But as far as getting the insects, that would be very cool. We have basically all the audio that would have the insects in the background so that could be looked at in the future, but we don't have like immediate plans to ID all the different ones. So, and then there's something else super secret, but we'll save that for the next one. Yes.
Okay. Oh, somebody asked, we would love to be able to pick up the Pinelands tree frog.
Cool.
So send us
we'll
an
look
email.
it up. Is that Pinelands, New Jersey?
Pinelands, one word. Yes.
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 Home
so there have to be
or something.
Home of what?
Nothing. Sorry.
Okay. So there have to be training data. We have to have recordings that we can create a net on those. So
we may already have it, but yeah. So we so there's a bunch of other things that have happened that we haven't really talked about, and you wouldn't notice. So for example, there were a lot of things that would false detect as Canada goose. So we've actually made a net that we run after BirdNet to try to reduce those false detections. And then similarly, we've made nets to try to improve detection of owls. So those things kind of happen and we don't really announce them. And so usually people don't notice if the false alarms go down, but hopefully they do. Although someone just emailed me this week saying the peacocks in LA are being picked up as Canada goose. So.
Peacocks.
So we're looking at that.
Imagine.
Yeah, so definitely doing the frogs is sort of high on my list because they're so tied to what's going on with their habitat and temperature, and they're very cool. So yeah.
Okay. And then someone asked just if are there collaboration spaces such as Facebook or other platforms where enthusiasts can collaborate, for example, different classroom sharing projects? So I would love to hear from you about those. We are definitely trying to expand our network and our collaborations and our community. So if you have things that you like to use already, we would love to hear from you or whatever ideas that you might have. And then one of our super fans, Colleen.
That's Colleen. Hi, Colleen.
Yeah. So our beta testers were colleagues. We forced into this, and some of them became true Haikubox lovers. And so she sent us a message that I will share with you, David, later. Just thank you, and yeah. #superfan. So unless there are any other questions, I think we're going to end on this note. Like I said, my plan is to get the recording up to YouTube this week. Again, we're a team of 4 people. So if you write to us and you don't hear from us right away, it's because we're doing many, many things. But really enjoying ourselves. So anyway, thank you so much for coming. And I guess that's it. If you have ideas for other webinars, let us know. Reach out anytime, really, with any questions or things you want to share. Okay? Thank you.
Great, thanks.